So, back to the talk. What is immunohistochemistry? So immunohistochemistry simply refers to the binding of an antibody to an antigen within tissues. So here you can see a paraffin-embedded human ovary tumour tissue, which is stained for PAX8, which is a common tumour marker. And IHC has been around for a while. It was first described in 1903 actually, but the first reported study was by Kuhn et al. In 1942, which used Ffpe labelled antibodies to identify pneumococcal antigens in infected tissue. So what are the applications of immunohistochemistry? So these can be used to diagnose disease, looking at histopathological tissue. So we can look at prognostic markers within cancers, and these can help identify any tumour-specific antigens and allows clinicians to define tumour as whether it is benign or malignant. It can also be used to diagnose tumours of uncertain histogenesis, for example, whether or not these metastases come from an unknown primary tumour. Also used in neurodegenerative diseases, so these images on the right here are TDP43, which is a marker in familial frontotemporal lobe degeneration. And on the left you can see this in a patient with this disease, and on the right you can see this in an animal model of the disease in mice. You can also look at IHC can also be used in identifying brain trauma. So staining for amyloid precursor protein following brain injury can help detect axonal injury as little as 2 to 3 hours following the head trauma. Other IHC applications, so for example we can use IHC in basic research. It is commonly used in basic research. For example, if you want to phenotype any genetic mutations, you can look at how the knockout of said mutation can affect any downstream targets, either within that tissue or in any effector tissues. And also with viral targeting confirmation, especially in things such as neuroscience, more and more viral targeting is being used to manipulate local gene expressions. These viruses are often tagged with GFP or similar tag. We can amplify these tags and then visualize whether or not the viral targeting has been in the correct location. And this was something that I used a lot in my own work. IHC is also really useful in drug discovery, as it can be used to up or down regulation of disease markers in response to certain treatments. So here we can see IBA1, which is a marker for microglia. Microglia are highly important in the CNS, in the brain, as they act as the macrophages of the brain. And when they become active, that is an indication of infection or stress on the brain. And here you can see that following ketamine treatment, these arrows are showing you a lot more of these microglia have become activated. So before we need to start our protocol, we have some things to consider first. So one of those is fixation and the type of fixators we use. Tissues and cells need to be fixed before staining, and this is to prevent cells and structures falling apart. Tissue requires either a long fixation process through immersion or through perfusion. Often it is good to try and perfuse with PBS prior to perfusing with formaldehyde, as this will remove any blood, and blood can contaminate various antigens and affect our staining. So formaldehydes work by cross-linking proteins, so formaldehydes such as paraformaldehyde or formalin, and they create covalent cross-links, essentially gluing these proteins together to form an insoluble meshwork. Formaldehyde is good because it conserves the cellular morphology. However, this cross-linking of proteins can mask epitopes and can also cause autofluorescence if using a fluorescent detection system. Acetone and other organic solvents act as fixatives as strong dehydrating agents, as these dehydrate cells and cause proteins to precipitate and denature in situ. Acetone is one of these and is good as it is can be used as an alternative to formaldehyde, as it is often gentle on epitopes. However, it can cause a loss of lipid components due to the nature of the fixation. Some other considerations that we need to take into account before starting is how we prepare our tissues. So tissues fixed by formaldehyde or formalin are often typically embedded in paraffin before sectioning, and these are called typically referred to as Ffpe sections, which mean formalin fixed and paraffin embedded. So when we have our Ffpe sections, we then need to, once they are firmly embedded within the paraffin, we then go on to cut these on the microtome. However, some fixation steps might not be appropriate for certain tissues, so instead we can cut these straight from frozen. So in order to do this, we will embed our tissues in a cryogenic protection such as OCT, which stands for optimal cutting temperature compound. So then we can cut these sections on a cryostat and then mount them onto slides. So the differences between cryostat, so frozen section, and paraffin Ffpe sections is that frozen sections are often good because they don't have their epitopes masked by some fixation, but you do get some disadvantages features such as poor tissue morphology and decreased resolution at higher magnifications. And another thing to take into account when we're preparing our samples is the thickness at which we cut our sections. So we want to ensure that our sections are cut at around 5 to 10 micrometers thickness. If this is too thick, then you will get strong signals and high background due to the fact that there's just simply more target antigen present in your tissue, gating more signal. If they are too thin, then they can be easy to damage and often difficult to embed as well. One thing to note as well when sectioning our samples is that we want to have well-coated glass slides. You can get very good commercially available, but you can also make these yourself if necessary through things such as gelatin coating. It's really understandably, we really want our tissue to adhere well to the slides, otherwise they can slip off and cause issues further down the line. Something else to take into account is if we need to do any antigen retrieval. Antigen retrieval is typically carried out before staining, as this is required in most formalin-fixed tissues, as this can potentially unmask any epitopes from our fixation process. So the options we have here are heat epitope retrieval. And to do this, we will heat our slides up either using citrate buffer, which is pH 6, or tris EDTA, which we can also often use for phosphode proteins. And another option is, which is less common, is proteolytic induced epitope retrieval. And to do this, we will use Proteinase K or trypsin. So here you can see a paraffin embedded tonsillitis tissue. Following different types of antigen retrieval, so these are both using heat antigen retrieval. So on the top here, you can see no retrieval. And then with tris EDTA, which is around pH 9, you can see a slight specific staining of our antigen. And then at the bottom, you can see that sodium citrate is the best choice for our antigen retrieval here. So what are the differences between the two? So heat-induced epitope and retrieval is where a slide is heated up in a microwave or a pressure cooker, often with a citrate buffer at around pH 6, or with tris EDTA, which has a higher pH. And as I mentioned before, it's good for antibodies against phosphatyrosine P. So proteolytic induced epitope retrieval is good if you have an epitope which is difficult to recover, and this is at neutral pH, use the Proteinase K or trypsin, and is incubated at around 37 degrees. Something to be aware of is that unequal heating could lead to unbalanced epitope retrieval and therefore affect any of your analysis further down the line. And if you're using P, then too strong enzymatic retrieval could also damage your tissue morphology. So this is a general protocol, and I just want to note that this is a very brief overview, and that this protocol will need to be tweaked based on your experiment needs, for example, your tissue type, your antigen abundance, the antibody you're using. And we have on our website product-specific protocols, which we highly recommend you following based on the antibody, as this will provide you with the best results that we have determined already in our labs. So, to start with, we need to remove the paraffin, because paraffin is a hydrophobic compound, and this will obviously stop any staining if it's remained on the tissue. So to do that, we use xylene. You can also now get xylene-free solutions that help do this as well, because xylene isn't the nicest compound. And following xylene, you then have to rehydrate your slides in decreasing ethanol solutions. Following that, we have a blocking step, and the blocking step is important to reduce any non-specific binding. And then we need to wash the blocking off, so washing steps are very important here before incubating with our primary antibodies. Concentrations and incubations based on either what the manufacturer recommends or on your own protocol that you know works. And then following that, we need to wash off our primary, so ensure that we're removing any unbound primary antibody from the system before incubating with our secondary antibodies. And in this example here, we would incubate with a HRP tagged secondary antibody before then visualizing this with DAB. And for example, when using a chromogen, when using DAB, we want to make sure that we are carefully timing how long it takes for a reaction to occur, so that we don't overstain or understain using this DAB reaction before stopping it. Then following that, we can either use a counter stain, so such as Hurst, which is a nuclear stainer, and then we need to mount and seal our samples. So we need to block our tissue And this is a really important step, the blocking step, as this prevents any non-specific binding of antibody to tissue or Fc receptors. Protein blocking typically uses normal serum, and serum is a common blocking reagent as it contains antibodies that bind to the non-specific sites. It is important that the blocking sera is not the same species that our primary antibody was raised in, otherwise we will lose our specificity of the secondary antibody. BSA is also another common blocking reagent, and this is often used in conjunction with Tritinex, which is a surfactant and will help wetting the coating of the BSA to our tissue. If using chromogens such as HRP or AP, it's really important that we block any endogenous enzymes that might be present in the tissue before we add our secondary and primary antibodies. So to do this, for HRP, you typically block with H2O2, and for alkaline phosphatase, you block with acetic acid or similar buffers. And as mentioned, this is really important as we want to block any endogenous enzyme activities and therefore prevent any non-specific activity which could give us some false positive results. When choosing our antibodies, there are lots out there, so it's really important that we do our own research before choosing any antibodies for example, new experiments or new tissue. So things to look out for when choosing our antibodies are the number of citations this antibody might have, specifically looking at whether or not this antibody has been used in the same tissue, the same experiment type that you have, so the same sort of like fixation and slicing and even species, for example. So these can all be found, for example, in our website. You can see where the antibody has been cited and what application and species it was used in. It's also highly important that we look at how the antibody was validated and whether or not that manufacturer has the original validation data, so that if you're having any issues, you can speak to this manufacturer and they can provide you with this information. We also want to ensure that there is validation data on the application that you require, so in this case, immunohistochemistry. And in general, we want to have a look at whether or not you need a polyclonal or a monoclonal antibody. And in general for IHC, polyclonal antibodies are most traditionally used. And just to remind everyone, a polyclonal antibody is one that has been raised against the whole protein or antigen, and it will therefore have higher affinity as there will be lots of antibodies that bind to multiple different epitopes. And so these are good for the majority of stainings. However, if you have an antigen which is highly abundant, then you might want to use a monoclonal antibody, and monoclonal antibodies have raised against just one epitope, and therefore they have much higher specificity and are less likely to cross-react. And finally, one more thing to look at when choosing your primary antibodies is whether or not you need to have it to a different host species to those that you already have. Say, for example, you want to do a multiplex staining experiment. The easiest way to do this is to either have your primary antibodies raised in different hosts, so for example, rabbit or mouse. So when we buy a new primary antibody, or we have testing it in a new situation, for example, a new tissue or a new species, you want to optimize this primary antibody in order to get the best results. So the way that we recommend you do this is to perform titration experiments, and this will help determine the best concentration and incubation time for your tissues and targets. If you have a high affinity antibody and highly available target, then you will want to use a low concentration. And this is the reverse. If you have lower availability targets, you might want to try increasing the concentration. Incubation time as well. If you have a high affinity antibody with high abundance, then you will only need a short incubation time, often at room temperature. If you have a target which is poor accessibility, this will need an increased incubation time, and this will help it to penetrate the tissue. So when we're choosing our secondary antibodies for immunohistochemistry, we really want to ensure that we will get the best signal-to-noise ratio, and this means that we reduce background staining. Another thing that we need to take into context is potentially the subclass specificity. So this is applicable if you're using monoclonal antibodies. So different monoclonal antibodies, primaries, might have different subclasses of isotypes. So you might have an IgM or an Ig2A subtype, and you can also get secondaries that will specifically target these different subtypes. So this might be good if you have two different primary antibodies raised in mouse, and if you know that if they have different isotype specificities, then you'll be able to use different secondaries in order to perform a multiplex staining. However, if you are unsure and you just want to target your monoclonal antibody, in order to ensure that it will recognize that subclass, ensure that your secondary antibody states that it is IgG heavy and light chain, and this will target all primary antibodies raised in that host. So another thing that we want to be aware of is any cross-absorption. So this means that we want to reduce any possibility of cross-reactions with other species. So for example, we want to try and avoid any mouse-on-mouse combinations. So that would be, for example, using a primary antibody raised in mouse that in mouse tissues. However, if this is your only option, there are some kits out there, mouse-on-mouse staining kits, that will reduce any background noise that you might see. Something else that you can be aware of are secondary antibodies that are Fab2 fragments. So these are missing the Fc chain here, and they are smaller and are therefore more easily able to penetrate tissue. You are also reducing any background signal through binding to any endogenous Fc receptors, and thus are good for tissue which has high Fc receptor concentrations, such as spleen and thymus. So now we've finished incubating our primary antibodies, we want to think about how we're going to visualize this. So you can either do this using chromogenic or fluorescent staining. Most chromogenic detection systems use HRP, so that's horseradish peroxidase, which will interact with DAB and forms a black-brown insoluble product, or alkaline phosphatase, which interacts with AUC, which forms a red product. And often these come with signal enhancers as well, such as the ABC complex and something else. So as well as chromogenic stains, you have fluorescent staining. So fluorescent staining is where we have a fluorochrome attached to our primary antibody as direct immunofluorescence, or attached to its secondary antibody as indirect immunofluorescence. And fluorescent staining is excellent for multicolor staining panels. So just going to quickly go over the difference between fluorescence and chromogenic stains. So fluorescent staining is excellent for multiplexing, as it allows you to image multiple targets just within the same sample. You also get better co-localization, and you can see protein-protein interactions with this fluorescent stain. You also have higher dynamic range with fluorescent staining, as this allows you to see highly abundant antigens and low abundant antigens at the same time. However, some of the negatives of fluorescent staining is that the staining will fade over time, and you also have lower sensitivity. It also requires more complex imaging protocols, because you require more complex microscopes, such as a wide field fluorescence or a confocal microscope, in order to see these. Chromogenic stains are more sensitive due to higher signal amplification. As long as the slides are stored properly, you will get long-lasting signal, essentially forever. You also have easier imaging, because you just need a bright field microscope for this. However, as mentioned, it is slightly worse for multiplexing and co-localization studies. You have a lower dynamic range, and also the protocol is ever so slightly long. So these are just different examples of fluorescent and chromogenic stains. So on the left here, we have an example of chromogenic staining, with DAB being used to visualize the EDCA adherent antibody in this breast cancer tissue slide. And on the right here, this is a fluorescent stain. And here you can see two stains, where in red we have our MAP2 antibody, which is a common neural marker, and in green we have our GFAP antibody, which is a common glial marker. And you can nicely see their distribution here. At Proteintech, we have recently launched our new range of Coralite fluorescent dye conjugated antibodies, and these are where our popular monoclonal and polyclonal antibodies are now available, which are directly conjugated with fluorescent dyes. And so this negates the need for a secondary antibody with a fluorophore, and therefore they act as perfect tools for your IF studies, which require any multiplex co-labeling and reduce the need for secondary antibodies, and therefore can speed up your experiments and save you valuable time. So moving on, we now need to consider what controls we will need for our experiments. And it's highly important that we run these controls at the same time as our experimental slides, in order to negate any differences that you have with day-to-day variability between experiments. So our antigen controls need to include a positive control, and this is where we stain a tissue which we know expresses the target antigen. And so this will confirm that our protocol is working, and will also validate any negative results, rather than if you're using new tissue and you can't see any antigen staining, you might think that the antibody isn't working or your protocol isn't working, but in fact if you use a positive control, then you'll be able to validate this. We also need a negative control, so this is where we have tissue that is known not to express target antigen, and this will reveal any potential false positives, or non-specific signal. Here on the right, you can see our HAD-HA antibody, which is used in cells which are expressing HAD-HA, and then on the bottom you can see, following siRNA of HAD-HA, you can no longer see this specific staining. And one further antigen control is just staining the endogenous tissue, so this is where the tissue is prepared in the exact same way as our experiment. However, we do not incubate with any primary or secondary antibodies, and especially in immunofluorescence, this will reveal any potential autofluorescence that occurs due to cross-link fixatives. We also need to look at reagent controls, and this is where we have no primary antibody, and this ensures that our signal is detected as specific for the target. We also need to test all our fluorophores or chromogens in isolation, and again, this ensures any background noise we're seeing is not specific to our target. We also it's also recommended to use an isotype control, and this is where you would incubate with a non-immune immunoglobulin of the same isotype and the same concentration as our primary antibody. And again, this is another way to ensure that our observed signal is specific. So lastly, just going to go over some quick troubleshooting queries. So what do you do, and what is the cause potentially if you have weak or no signal? So one cause is that your primary or secondary antibody has lost activity, and our suggested solution here is just to use either a new lot of antibody, and also to check the storage of your antibody, and always ensure that you're following your manufacturer's instructions rather than just going with whatever you have done before. So for example, at Proteintech, we store our antibodies with glycerol, therefore you do not need to allocate. Another cause is that your antibody conditions might not be fully optimized. So one way that we can do this is to titrate our antibody concentrations and adjust incubation times accordingly until we potentially see some actual decent signal. One issue is that you might not have your protein of interest expressed, so as mentioned, always run a positive control. If your protein of interest has low expression, then try signal amplification steps. And if you have a damaged epitope, then this could be due to poor epitope retrieval or poor fixation, so try a different epitope retrieval technique or a different fix. Another common troubleshooting query is if you have high background or non-specific staining. So this could be if your primary or secondary antibody concentration is too high, so just try a titration experiment. If you have any non-specific antibody binding, you might want to try prolonging your blocking step, increasing your concentration of your blocking solution. It could also be due to poor sample washing. So as mentioned, it's really important that you have thorough washing steps following blocking, antibody application, primary and secondary, as this removes any unbound antibody in the system that could be causing further signal, especially the high background signal. You also can get high background due to damaged epitopes, so as mentioned before, try a different epitope retrieval technique or fixative. And lastly, if you're seeing inappropriate morphology with your results, this could be due to harsh antigen retrieval conditions, so you will want to optimize buffers and your temperature and pH. You might have unclear tissue structures, so this could be if you're using the wrong section thickness for your tissue. Your protein of interest might not be expressed, so try running a positive control to test your experiment conditions. And if your tissue is not sticking to the slides, then this could be causing it to bunch up or fold over, which again would quite obviously cause inappropriate morphology. So for this, we recommend using coated slides and ensuring that you have optimal fixation and epitope retrieval that isn't affecting your tissue adherence.